Hi, this is Michaela Fenwick doing my DNA extraction lab. The first question asks what DNA is and what its role is in living organisms. DNA is a complex molecule. It is made of nucleotides that all have a nitrogenous base, a phosphate group, and a sugar, either deoxyribose or ribose, deoxyribose for DNA, ribose for RNA. So the beginning of this lab, I'm going to go ahead and I've got my peas in water and I'm going to add some salt. The purpose of the salt is to neutralize negative charges and allow the DNA to stick together. So I'm going to go ahead and do that. Here I've got my peas in water and I'm going to add some salt to that. And then I'm going to go ahead and blend. Pretty good. I'm going to blend it a little bit more. All right. Now I have my blended peas and salt water. And I'm going to strain that through a cheesecloth to remove the solid particles. And I'm just going to use this stir to kind of ease things through. As you can see, all the solids are kind of gathering here at the top. All right, I've strained all of the solid elements out and now I'm left with this pea soup looking stuff. I'm gonna add some detergent now. The detergent just destroys the phospholipids in the plasma membrane and allows the cells to open up and also the nucleus to open up so that we can see the DNA. So I'm going to stir that and then let this chill for 10 minutes. So my solution is done being chilled and I'm going to move on to the next part of the experiment, which is to pour some of the liquid into a test tube so that we can do our DNA extraction. I'm going to pour this over the sink. All right, here I have my pea soup ready to go. I'm going to put in the papain enzyme. This is an enzyme to destroy the protein coat of the DNA, which exposes the strands. And I'm just going to gently stir that. And then I'm going to get my alcohol out of the freezer. alcohol is used to help separate out the DNA because DNA is not soluble in the alcohol. Cooling the alcohol down slows the speed of the enzyme degradation of the DNA so that we can actually recover more of it. So I've added about the same amount of alcohol, and now we'll wait for the DNA to precipitate out. All right, this is the result of my DNA. You can see the DNA. Hopefully you can see it kind of floating to the top. The DNA is pretty viscous. You can see that it's kind of sticks to the side there. There's not as much of it as I thought there would be. I remember doing a strawberry and I felt like there was a lot more DNA than this, but the DNA of the strawberry is polyploid, or the strawberry is polyploid, so maybe that has something to do with it. Or maybe there were errors in my procedure, but you can definitely see the DNA. So that's a good result.